Hey guys, so this is chapter 6, Sensation and Perception. This is one of the more difficult chapters just because it is so visual and conceptual, and so it's hard sometimes to answer questions in words over concepts that are more easily seen. So just be aware of that and pay attention to the wording that I use on things because you'll be asked questions in words rather than in images. Okay. So the first thing that you want to know the difference between in this chapter is sensation and perception. A sensation is something that you physically feel, taste, see, smell, hear. So it has to do with your senses. So a sensation is something that you feel. Whereas perception has to do with your interpretation or understanding of something. So generally, we feel a sensation and then we attach a perception to it. You feel a pat on your shoulder and then you think to yourself, oh, who is that? Who's trying to get my attention? Or what is that? Kind of a thing. So the sensation is just the feeling of someone tapping you on the shoulder. The perception is, who is that? What's going on? What do they want from me? How you respond to and how you think about the sensation. And then there's bottom-up processing and top-down processing. With bottom-up processing, you start with a sensation and then you work up to perception of that sensation. So we start looking at the individual details of something. We use them to see the whole picture, to perceive an understanding of something. And that's how bottom-up processing works. So we start with the sensory receptors, work up to the brain's integration of that information, their interpretation, integration, perception. Whereas top-down processing is when we first approach something with our perception, our higher level mental processes, and then we work to break that down into smaller pieces and pick out the details from there. So bottom up, starting at the very basis, the sensation of what something is, and working up to perception, versus top down, starting out with your perception, your opinion, your interpretation, and then getting down to the details of what build that perception. So bottom up, sensation to perception, top down, perception to sensation. Okay, this term transduction is really important. And all that transduction means is that we are converting some type of energy from one form to another. So if you've ever taken physics, you probably have talked about, how mechanical energy converts into heat energy and those sorts of things. And that's kind of the same idea here. We're going to be talking in this chapter a lot about vision and hearing and stuff like that. And so when we talk about transduction, we talk about how a light wave is transduced, is changed from light energy into activity that is electrical activity that's actually going on inside of our brains. So that's how transduction will really work in this chapter. And then when we get to, auditory stimuli, hearing, then that's the conversion of sound waves into electrical energy that we can then perceive in our brains. So just be aware that transduction has to do with changing from one form into another form, one type of energy into another type of energy. Okay, the absolute threshold and the difference threshold. This is really important. So when we're talking about, remember when we were talking in chapter 2 about thresholds, and that's like the minimum amount of stimulation that a neuron needs in order to be able to fire an action potential. Well, absolute threshold in terms of sensation and perception is the minimum amount of stimulus energy necessary for us to know that something is present and to detect that something 50% of the time. So we have an absolute threshold for our hearing. If you've ever in elementary school had to do those hearing tests where they bring you in and they hook you up to, or they give you these headphones and hook you up to this device where every time you hear a beep, you press a button. Well, you're not going to hear some of those beeps because they are so quiet that they are below your absolute threshold for recognizing that there is a sound there at all. So your absolute threshold is how much, in this case, sound do I need? At what volume does the sound have to be in order for me to notice that the sound is there at all? If we we're slowly diffusing, let's say, cologne into a room. How much cologne needs to be diffused before someone's going to go, oh, it smells like something, right? That's your absolute threshold, that moment of detection. Hey, it smells like something, or it sounds like there's a noise, or that sort of thing. Whereas the difference threshold is a different idea. So with the difference threshold, you already know that the stimulus is there. You already hear a sound or see something or feel something or smell something. But the difference threshold is how much does that stimulus have to change in order for us to realize that it has changed. So if, I'm playing music at volume level 15, if I, without telling you, turn it up to volume level 16, you're probably not going to realize that I changed the volume at all because it's below your difference threshold. The difference between volume level 15 and volume level 16 isn't a big enough difference for you to be able to detect it. So, maybe I turn it up to 17, I turn it up to 18, I turn it up to 19, right? Slowly. And then at some point you say, it sounds like that music got louder. That point where you say, it sounds like it got louder, it sounds like the stimulus changed, well, now we've reached the difference threshold. And so what Weber's law states, Weber's law is crucial too, and this whole slide is really important. What Weber's law states is that in order to recognize that there has been a difference that has occurred, the stimuli that you're being exposed to has to change by a certain percentage, not just by some arbitrary amount. So let's think about it like this. If I have my eyes closed and I'm holding 10 pounds in my hand, and you put a pound on top of it, I may or may not notice that that pound is there. But let's say you put 5 pounds on top of it. Well, I'm certainly going to notice 5 pounds. That's a bigger difference. Now let's say that I was holding 100 pounds. If you put even 5 pounds on that, well, I'm probably not going to realize it because I'm already holding 100 pounds. What's 105? But if you add 50 pounds to it, well, now I might realize that something has changed, right? So even though 5 pounds was enough for you to notice a change when you were holding 10 pounds, it's not enough for you to notice a change when you're holding 100 pounds because the two stimuli don't have to differ by some arbitrary amount like 5 pounds, but rather by some minimum percentage. So when I was holding 10 pounds and I had 5 pounds added, well, that's a 50% increase to my weight. Versus if I put 5 pounds on top of 100 pounds, that's just a 5% increase to the weight. I may not notice it. But if I increase that to 50%, I'm putting 50 more pounds on top of my 100, now I will notice it. So Weber's law states it's not a minimum amount, but a minimum percentage that the two stimuli have to differ by in order for you to reach the difference threshold. Okay. Signal detection theory really just states that whether or not we notice a stimulus is present is influenced by all sorts of these elements of our psychological state. Our experience, our expectations, our motivation, and our alertness. So let's use the diffusing cologne example again. If I tell someone, hey, in a minute, you'll probably start to smell a cologne. Well, now they're going to smell it way sooner than they would have if I hadn't have said anything. So because I told them what to expect, I actually lowered their absolute threshold for that thing. Because now they're on high alert thinking, do I smell anything? Do I smell anything? Do I smell anything yet? Do I smell anything yet? Oh, I think I do now. Yeah, I smell it now, right? But if you wouldn't have told them anything, how long would it have taken before they did actually start to smell it, right? So signal detection theory, the main thing that you want to write down for this is that this is saying that our absolute threshold can change. Our absolute threshold is not held constant and it's just some arbitrary level. Our absolute threshold is absolutely influenced by our expectations, our motivation, how alert we are, right? All sorts of other factors. So signal detection theory just says, our absolute thresholds can change in response to environmental factors. Priming is when you sort of bring an association into someone's mind without them being aware of it. So have you ever maybe met someone and immediately like had a really good feeling about them or a really bad feeling about them? If you did, it was probably because they primed you somehow. Usually, if someone looks somewhat similar to someone else that we know and we like that person, we automatically start to like this person too. But if they remind us of someone we dislike, we actually start to dislike them as well. And that's priming. We are bringing an association forward, an association between this new person and someone that you've known previously. And you may not consciously be thinking, hey, this person looks exactly like my friend who I really like, or exactly like my enemy who I really hate. But regardless, we are triggering that association in your mind and therefore causing you to be primed. Another example is when we put people in a lecture hall and we give them a lecture about rabbits, bunnies, versus people who we've given a lecture about, anything else. And then at the end, we ask them to write down a series of words, and one of the words is hair. The people who listen to the bunny lecture at incredible rates will spell hair H-A-R-E, versus the people who were given some other kind of lecture are way more likely to spell it H-A-I-R. That's an effect of priming. You probably weren't aware of it. You weren't thinking, I better spell hair like the rabbit since I was just listening to a rabbit lecture. No, but because rabbits were on your mind, which can be called hares, H-A-R-E, it primed you to then spell the word hair in that way. So it brought up some association that predisposed your perception, memory, or response. And again, this is most of the time unconscious. And then we've got this idea of sensory adaptation. This is pretty easy. If I put a hair tie on my wrist, I don't feel it there for the rest of the day, right? You only feel that hair tie on your wrist for a couple minutes and then you don't feel it anymore. Same thing with, let's say, putting a sock on your foot. At first, I feel that sock there. It might be kind of annoying, right? But after a few minutes, I no longer feel the sock constantly on my foot. And that's the idea behind sensory adaptation, that if we are constantly stimulated by something, then our senses adapt to that feeling and we no longer feel it. And so the main idea here is that our sensory receptors are wired to be attentive to new stimuli. And so if we bore them with constant stimulation to the same thing, that's going to lead those sensory receptors to free up our attention for other things and to not constantly transmit the sensation to your brain. So what I want you to write down on your worksheet is that sensory adaptation leads the body to ignore irrelevant sensations. It leads the body to ignore irrelevant sensations. And this is also quite similar to selective attention. Back from chapter 3, when we were talking about selective attention, we're talking about, tuning things out that are visually unimportant in order to free up our attention for the things that are important. Well, this is the same idea. We're freeing up our sensory receptors' attention from an unimportant sensation, a constant unimportant sensation, so that they're able to perceive new, novel stimuli as they come along, because those might be more important. Okay. Your perceptual set is simply just the experience that you bring with you into a given situation. So it's just your set of tendencies, assumptions that affect what you hear, taste, feel, and see. So everyone has their own perceptual set. It's just based on the experience that they're bringing in to any given situation. So your perceptual set is really just your predisposition to see, hear, feel, experience something in a given way. So essentially, what we expect to hear, see, or feel influences what we actually hear, see, or feel. But what factors might influence our interpretations and how? Our perceptions are going to differ based on the context. A good example, when we drive, we hate the pedestrians and the bikers. But when we're walking or biking, we hate the drivers, right? So it's based on the context that you find yourself in. You're always going to have sort of a self-serving bias and think that you're doing the correct thing and it's other people who are doing wrong. Our motives can also put bias on our interpretations, so our motivation. For example, a hike doesn't look super daunting when you're at the beginning because you're energized and you're motivated, but once you get going and your motivation declines, well, now it seems like this is a much longer and effortful task than we ever thought it was before. So our motivation can bias our interpretations. And then our emotions have a huge impact on our perceptions. We draw out different meanings in neutral stimuli based on what we're already feeling. So if you're already feeling sad and upset, and someone who regularly says hi to you in class says hi to you as usual, but you think that maybe it feels a little bit disingenuous, or you don't think that they were, being very kind to you or something like that, right? That's all because of your emotions. You were already having a bad day, so that affected your perception of how your friend was interacting with you or your classmate. Okay. So now we're moving into vision. So hue has to do with color. Hue is just synonymous with color. So the hue of a light is what we're going to call red, blue, green, orange, those sorts of things. It's just hue is color. And know that hue is determined by wavelength. Hue is determined by wavelength. And so you may know this from other science classes, but red light, so if you're on the spectrum, Roy G. Biv, right? Red, orange, yellow, green, blue, indigo, violet. Red has the longest wavelength, whereas violet has the shortest wavelength. So that wavelength being the distance between two peaks, right? So from here on red to here, that's a very long wavelength, versus over here for violet, here to here, very short wavelength. Intensity is the amount of energy in a light wave or sound wave, which influences what we perceive as brightness or loudness. So the more intense a color is, the brighter it is to our eye. The more intense a sound is, the louder it is. And intensity is determined by the amplitude of the wave, meaning the height. So if we look over here on this graph, this would be considered the amplitude. And we can see it both above and below. Amplitude determines intensity. So a very bright light would have a very high amplitude, whereas a dim light would have a very low amplitude. And then your retina is what is light sensitive in your eye. So the retina is really what's going to be determining hue and intensity here. It contains receptor rods and cones, plus layers of neurons that begin that processing of visual information that we're taking in. And then accommodation is this idea that the eye's lens will actually change shape in order to focus near or far objects on the retina. So if you have an issue with accommodation, the solution to that is contact lenses. Notice contact lens, because it's the eye's lens that needs to change shape in order to focus on near or far objects. So that's why we use, false lenses, because your eye lens is not doing the job it's supposed to do in order to get you to focus on near or far objects. So we correct it with a contact lens, which will do this job instead. Okay. You absolutely want to know the difference between rods and cones. And so this I need to see written down on your worksheet. Next to rods, write down black and white and peripheral vision. Peripheral. I don't know how to pronounce that super well. Is it peripheral? Peripheral? I think it's peripheral. So next to rods, black and white and peripheral vision. Next to cones, write down color and fine detail. Color and fine detail. So cones, color. Cones, color. The C's go together. Whereas rods are black and white and peripheral vision. So your rods are going to be on the outside of your eye, whereas your cones are clustered all in the middle of the eye. Again, because rods are for peripheral vision, whereas cones are for central vision. So your rods are going to be on the outsides of your eyes, and they're going to detect black, white, and gray, and they're really sensitive to movement. Now, this is adaptive. The reason that we have rods and that they serve the function that they do is because when our ancestors were alive and it was very dark outside, you need to be able to see movement, in case a predator is coming along. And so that's what the rods are for. They're for contrast between black and white, and then they're also very sensitive to movement, so that even if you don't have bright light to see through, you can still detect movement because of your rods. They're hypersensitive for that. And so this is necessary for peripheral and twilight vision when your cones really can't respond because there's no color or light. And then your cones are concentrated, like I said, near the center of the retina, and they function in daylight or well-lit conditions. They detect fine detail and give us color. Okay, when it comes to the optic nerve, this is the nerve that carries light information from our eye to our brain. But what you really want to know is that the blind spot is the point where the optic nerve leaves the eye. Your blind spot is the point at which the optic nerve leaves the eye. And so if you're staring straight forward and you bring your finger from in front of your face all the way over past your ear on the side of your head, your blind spot is the point at which you stop being able to see your finger, like the point at which your eye can no longer see back that far. That's your blind spot, and that is also the point where the optic nerve leaves the eye. No receptors are located there. Too far off to the side. And then the fovea is the central focal point of the retina, and you definitely want to know that all of these cones lie within the fovea. You have a cluster of cones in your fovea. Your fovea is the central point. I told you that cones are in the center of your eye, so this is where all of the cones cluster together. Okay. There's two theories of color processing. The first is the Young-Helmholtz trichromatic color theory, and this theory says that there are 3 types of color receptors in the retina. One that goes with red, one with green, and one with blue. Now, the thing that people miss most on test questions is they want to say red, yellow, and blue. Don't make that mistake. It's hard to figure out exactly how you can create yellow from these colors, but nonetheless, this is the theory, that we have color receptors for 3 colors, and those colors are red, blue, and green, not yellow. So just memorize that it's those 3 colors in the trichromatic or 3-color theory. Now, opponent process theory is a little bit more difficult to grasp. And so it's saying that there are opposing retinal processes that enable your colored vision. So some cells are stimulated by green and inhibited by the color red. Some are stimulated by red and inhibited by the color green. And so what you want to know from this theory is which colors pair together. Know that red goes with green, blue goes with yellow, black goes with white. And this really has to do with after images. So if you've ever, stared at something for a really long time, let's say you stared at something red for a really long time, when you look away somewhere else, you get a green flashback of that image, right? If you stare at the sun for too long, that's yellow, and you look somewhere else, you get kind of a blue or purple after image. And that's what opponent process theory is talking about, is this after image idea. If you stare at this flag for long enough, right in this little center point right there, and then you quickly shift your eyes over to the blank space on the right side, you should see a correctly colored American flag, because green will turn red, black will turn white, yellow will turn blue. But the idea here really is if you're staring at something red for super long, then those red processors tire out. And when you look away and they go to rest, you get flashback from the other end of that receptor. And so the other end is green. So the flashback image that you get is the opposite color as the one that you started with. Okay. Feature detectors are here to respond to specific features of a stimulus. Like you have a feature detector for detecting shape. You have a feature detector for detecting angle. You have a feature detector for detecting movement. They're specialized cells that respond to specific features of any given stimulus. And so feature detectors are what is going to enable us to be able to parallel process things. So we talked about this way back in chapter 3. But parallel processing is being able to process many aspects of something simultaneously. So like processing motion, form, depth, and color of a bird all at once, and then integrating that information into a perception, which is, oh, this is a robin or hummingbird or whatever. And so you'll have a feature detector for detecting motion. You'll have a specific feature detector for detecting form, a specific feature detector for detecting depth, a specific feature detector for detecting color, amongst other things. So feature detectors enable parallel processing. Okay. Gestalt psychology. So anytime you see the word gestalt, you should immediately think whole, a whole piece of something or a whole element of something. So Gestalt psychology is all about organizing things into wholes, whole concepts, whole pieces. So here, when you look at this picture, if I were to ask you, what is this? You'd probably say, it's a soccer ball. Well, not really. What it is truly is just a series of tilted pentagons, right? But because your brain is sort of filling in these gaps and saying, well, this should be connected here because of the form and the angle of these shapes. And so your brain actually organizes it into this whole picture of a soccer ball, right? So that's this idea here. In perception, the whole may exceed the sum of its parts, meaning that you see more than what is actually there because your brain has filled in those gaps in order to make a whole thing that you can understand a little better. A whole perception. Another example here, a lot of people will see two triangles, one upside down and one right side up, but really what's here is just 3 V's. There's not even one triangle, but your brain fills in those gaps to make a gestalt, an organized whole of something. Figure ground are these cool illusions that we have where if you look at the white space, you see some kind of maybe candle or something in here. If you're looking at the white space. But if you're looking at the dark space, you see a face on this side and a face on the other side. So that's figure ground, is really when you're asking yourself the question, is this the figure and all of this is the background? Or is it the other way around where this is the background and these are the figures? So it's a visual illusion where you can sort of shift your perception to see one thing as the main picture with background around it, or shift your vision so that the background becomes the main picture and the original main picture becomes the background. Figure, ground. What's the figure? What's the main picture? What's the ground? The surroundings, the background. And then there are 3 grouping principles. We have proximity, continuity, and closure. So when asked what people see here in this first image, they would say, well, I see 3 groups of parallel lines. But there's no reason to group them together. All that really is here is 6 lines, although, they are closer to one another in some places versus others. But by using the grouping principle, proximity is why you see like 3 tubes or 3 lanes instead of just 6 lines, which is truly what it is. It's just 6 lines. And then continuity is really just when you essentially connect the dots of something in your mind. So you perceive this as a continuous X instead of just a series of dots. And then closure, you would say that this is a circle and this is a square, even though they're not fully closed off. So in proximity, we group things together based on how close they are to one another. In continuity, we group individual pieces together if they look like they could form some type of shape if put together. And then in closure, you're just filling in some kind of a gap in a picture. So, both of these examples with the soccer ball and the triangle are examples of your using of closure. Okay. Depth perception has to do with being able to tell how far away or how close an object is to you. And we would use these studies called visual cliff studies, where we would actually put infant children or very small animals onto a glass table, onto a table with a piece of see-through glass beyond it. So we'd have some kind of table that we put this little baby or this animal on. And then we'd have a piece of glass. And they could walk on this glass and be fine, but beneath it would be all of this floor. And so what we do with the visual cliff studies is we'd have, the mom or the pet owner stand over here and try to coax the baby or the animal to come to them. Come over. And then the baby or the infant animal would crawl forward. They would look down and see that that's a big drop. And so if they would back off and they wouldn't climb across the glass, we would assume that they had depth perception, that they could tell that there was a drop beyond the place that they were at. And if they were to crawl forward, they would potentially be in danger of falling. So we would use the visual cliff in order to test for depth perception in infants and young animals. Mostly, just know that the visual cliff was used to test depth perception. Okay, when it comes to monocular and binocular cues, mono means 1, bi means 2, right? So a monocular cue is a depth cue that you can see with just one eye alone, whereas a binocular cue is something that you need both eyes to be able to see depth as a result. And so a couple examples for the monocular cue are interposition, which is basically just like a collage. If I put something behind something else, I only need to look at this with one eye to be able to tell that something is deeper than something else, that this one is behind this one. This is in front, so this must be, farther back at a greater depth from the eye. And then binocular cues, you need both eyes to tell what's going on. And the biggest binocular cue that we're interested in is retinal disparity. This can be a tough one for people to understand. So I'm going to draw a little picture up here in the upper right. If this is your face, and then you've got your right eye and your left eye. These are very spread out, I understand. But let's say we're looking from bird's eye view, that's your face and your 2 eyes. So think if you put your finger right here in front of your face, and you can do this while you're watching the video, if you put your finger right in front of your nose and you close each eye, one at a time very quickly, you can kind of see your finger moving from place to place, right? As I close and open each eye, it moves back and forth a whole lot. But if I move my finger way out in front of me and I do the same thing, it moves very little. The 2 images from your 2 different eyes are still different, but they're not quite as vastly different as when that object is very close to you. Because if we're thinking about, the angle at which your eyes have to be looking at this image, the closer that you get to your face, the more different the images that your two eyes see are going to be, because they're looking at that object at such different angles from one another, versus looking at an object so far away, there's hardly any tilt to having to to look forward into this object. And so the images that your two eyes are getting are approximately the same image when it's far enough back. But when something's really close to your face, your two eyes are getting very different images because they're oriented at different angles to be able to look at the image. So the idea here is that the greater the disparity, because we're talking about retinal disparity, the greater the disparity, or the greater the difference between the two images of the same object you see, the closer that object is to you. So this is this example. Let me change colors. This example right here. The greater the difference, the greater the angle too, the greater the difference, the closer the object must be to your eye. But the farther away something moves, the less difference we get between those two images, which is why your finger moves less when you're looking at it from a distance than when you're looking at it super close to your eyes or your nose. So really, just remember, the greater the difference, the closer the object. But the less difference, or the less disparity, the farther the object. This is kind of difficult stuff to talk about and illustrate in just words, but hopefully you get the general idea, and you can always ask questions in session if there is something that you didn't quite understand. Okay. The phi phenomenon is really just the perception of movement when there is no movement. So if you've ever seen one of those neon signs that lights up, letter by letter or something like that, but in your eye it looks like there's movement, it looks like it's moving from left to right, but really each letter is just lighting up on its own. So that's the phi phenomenon, is when you have this illusion of movement that happens when two or more lights blink on and off in quick succession. Perceptual constancy. This is a cool one. This is perceiving objects as unchanging, having some constant or consistent color, brightness, shape, and size, even when the illumination and the retinal image does change. So you would probably say that this square is the same color as this square, even though the lighting is different. But what you're definitely not going to say is that this square is the exact same color as this square. Well, as it turns out, if I cover up this picture piece by piece, those are the exact same color. Those are the exact same color. So perceptual constancy is us not recognizing that, knowing that yes, this is a little bit darker because of a shadow, that exists, but certainly not this dark. Certainly this is closer to this color than this one, right? But we're wrong, right? Turns out they're the same color. We perceived A as being consistently lighter than B, but they're the same. And then we have perceptual adaptation. This is the ability to adjust to some kind of change in your sensory input. So if you've ever put on drunk goggles, for example, that inverts your visual field or tilts your visual field, right? Well, if you leave those on for long enough, that will become your new normal, and you'll actually be able to navigate the world perfectly with drunk goggles on all the time because of perceptual adaptation. Our senses adapt to, or accommodate themselves to the circumstances that we're in, which is why when people lose an arm, like to a shark attack or something like that, they often can figure out ways to use only one arm to live their life. Or if someone goes blind in one eye, they figure out ways to use their one good eye to navigate the world around them. And that's because we have perceptual adaptation. We adapt to the changes in our perception. Okay, now we're into hearing, which should kind of be the last thing we cover here. So audition is just the sense or act of hearing, very basic. Pitch has to do with how high or how low something is, and that is dependent upon the frequency. So these are things you're going to want to know, like when we were talking about color, the fact that intensity depends on amplitude and hue depends on wavelength. You need to know these things. Pitch depends on frequency. Okay. And then we want to know about the middle ear, the cochlea, and the inner ear. So you want to know that inside the middle ear is where we have these three bones, the hammer, the anvil, and the stirrup. And that the middle ear exists between the eardrum and the cochlea. And then we have the cochlea, which is a bony, fluid-filled tube inside your inner ear. And your cochlea is really what's responsible for transduction. It brings in those sound waves and it converts them into electrical energy that your brain actually interprets as a sound. And then in the inner ear, the innermost part of the ear, we have the cochlea, the semicircular canals, and the vestibular sacs. So definitely know which of these things are in which section of the ear. This is kind of basic anatomy, but it is something that you do need to know for this exam. And then we've got two types of hearing loss. Sensory neural hearing loss is when your receptor cells get damaged. And by receptor cells, we really mean hair cells. Your hair cells. The hairs inside your ear. If you damage those, there's not much that you can do. So if you listen to music that's too loud, it will damage those receptor cells, those hair cells on the inside of your ear. And if you cause damage to those, they don't fix themselves. They don't fix themselves. And this is called nerve deafness. Conduction hearing loss has to do with some damage to the actual sort of mechanical or computerized part of your ear that participates in transduction. So the mechanical, mechanism inside of your ear that converts waves into neural impulses that tell you what something sounds like in your brain. With conduction hearing loss, there's an issue with that internal mechanism that participates in transduction. And then we have a cochlear implant. Just know that cochlear implants can help to treat sensory neural hearing loss, but there's really nothing that we have that helps to treat or manage conduction hearing loss. So know that the cochlear implant can be used to help repair not even repair, but bypass the hair cells in the first place that are damaged, and give you the sensation of hearing even though you do have damage to those hair cells, nerve deafness. So the cochlear implant can manage symptoms of sensory neural hearing loss, but it can't cure it. Okay. Now there's two different theories for how we hear pitch. So know that these are your two pitch theories. The first theory is place theory. And this is the idea that, let's say that this circle is your basilar membrane. The idea is that maybe this part of the basilar membrane is for, very high pitches, extremely high, like almost ones that we can't hear. But this section of the basilar membrane is for, pitches that are towards the middle of the scale. And this section is for the lowest pitches, right? So this idea that there's a specific place on the basilar membrane that gets stimulated, which then tells our brain, oh, well, if it's this place that got stimulated, well, then we must be hearing a very high pitch, right? And so when you hear a high pitch, it's because, this theory says, it's because a certain place on your basilar membrane was stimulated, and so your brain interpreted that place as being correlated to high pitch or low pitch but just know that it's about a specific place on the basilar membrane that determines what pitch you hear. Versus frequency theory. Frequency theory says, well, high pitches have a much higher frequency than low pitches. They have a very low frequency, very low wavelength, right? Long wavelength. And so the idea is that whatever frequency the sound that you're hearing is, makes your entire basilar membrane vibrate at a certain speed. If it's vibrating at a really fast speed, then your brain interprets this as a high pitch. If it's vibrating at a really low speed, your brain interprets this as a low pitch. And so we don't really know which, if either of these is correct, but the idea is that place theory says a specific place on the basilar membrane is stimulated by sound, and that determines our understanding of pitch. Versus frequency theory says, no, the sound itself causes the whole basilar membrane to vibrate at some kind of frequency, and whatever that vibrational frequency is will determine how we hear pitch. Okay, I'm going to try to move past these pretty quickly, because I know this is already becoming a longer video than the others. But influences on our experience of pain. So pain is totally a sensation thing, right? When it comes to biological factors, we have nociceptors, and those are receptors that are dedicated to detecting things that are harmful to us, harmful temperatures, pressures, or chemicals. And then your experience of pain partly depends on the genes you inherited, like your pain tolerance of your parents, and your physical characteristics. And then there's this idea of gate control theory. So this is the idea that your spinal cord has essentially a gate that can block pain signals or allow those pain signals to pass on to the brain. And so what you really want to know about this theory is that the gate is opened by pain that comes through small nerve fibers. So small pains are actually what trigger this gate to open so that we can feel the sensation, versus activity in large fibers, such as, let's say, getting a massage, actually shuts off that gate and does not allow pain signals to pass to the brain. So this is why, when you're getting a massage, yeah, it can hurt when they press down in one specific place, right? Because that's small nerve fibers that opens up our gate so that we can experience pain. But if they're massaging a large part of you, it's actually not painful, it's pleasurable, right? And that's because they're producing activity in a large amount of space or large fibers, which closes off that gate that tells our brain that we should be feeling pain. Psychological factors, our perception of pain is influenced by the amount of attention we focus on it. So distraction really is a cure for pain to some degree. And then our perception of pain has to do primarily with how much pain was felt at the peak of an experience and how much pain was felt at the end. So if we do experiments where we administer some kind of shock or something like that to a person at the start of the experiment versus at the end of the experiment, they tend to remember it being more painful if it happened at the end of the experiment, even if the shock was the exact same intensity for both people. The people in the sample who experience the shock at the start of the study have time to forget about it, and so by the end they're like, oh, it wasn't really that bad. But people who experience it at the end, they have a fresh memory of it, so they say that it actually hurt more than the people who were in the other group. So our perception of pain primarily has to do with how much pain we felt at the peak, at the worst part of the experience, and at the end of the experience. Social cultural factors. Pain is a product of our culture. It varies with social situation and cultural traditions. And when people feel empathy for another person's pain, their own brain activity partly mirrors the activity of a brain experiencing pain. And this goes back to those mirror neurons that we talk about in Chapter 7, Observational Behavior. or observational learning rather. Okay. What factors and methods can contribute to controlling pain? Well, placebos, because placebos work if you believe that they will. Believing that a placebo is going to work actually triggers your brain to release endorphins, and endorphins alter your perception of pain. So if you just believe that you've taken something that's going to help manage your pain, your brain releases endorphins and it actually does manage your pain, but not because of the placebo, because of your belief in the placebo, which caused your brain to release endorphins in the first place. Like I said, distraction is honestly a cure for pain to some degree. If you draw your attention away from a painful stimulation, then you can activate brain pathways that actually inhibit pain and increase pain tolerance. And this goes into hypnosis. Crazily enough, there are actually some people who can go through an entire surgery with no sedation or pain medication whatsoever, just under hypnosis. Now, that's not most people, of course, because most of us can't just dissociate that easily from our bodies and our brains, but some people truly can undergo an entire surgery under only just hypnosis. But so hypnosis is a social interaction where one person, the hypnotist, suggests to the subject that they're going to experience certain perceptions, feelings, thoughts, or behaviors. And dissociation just means a split in consciousness. So when we're talking about hypnosis, we're talking about dissociation, a split between the brain and the body. And then a post-hypnotic suggestion is just something that the hypnotist says that the person will do after they're out of hypnosis. And so, if someone goes in for hypnosis to help them kick their cigarette smoking habit, the post-hypnotic suggestion would be, once you come out of hypnosis, you'll feel disgusted anytime you see a cigarette, or you won't feel, the impulsive need to smoke a cigarette anymore. It's just some kind of suggestion that you're supposed to carry out after hypnosis is done, post-hypnosis, post-hypnotic. Okay. Know the 5 basic tastes. Umami is probably the one that you haven't heard of before, but we've got sweet, salty, sour, bitter, and umami, which just has to do with proteins. So umami is, I guess, the flavor that you get when you eat meat. Very strange. Olfaction is your sense of smell. These are some fun facts about that. Everyone has their own unique, identifiable chemical signature and scent, except for identical twins, because they are genetically similar 100%. Our perceptions and reactions to smells depend on our unique experiences with them, and scents are strongly connected to memories. This is because there's a hotline that runs through the area of the brain that controls information received from the nose, and then there are limbic structures associated with memory and emotion that also run alongside this hotline. So basically, the area of the brain that receives olfactory information is very close to the part of your brain that is associated with memory and emotion. And so that's why we get our strongest, most emotional memories triggered by certain smells, because those two parts of the brain are just very close to one another in proximity inside your head. Okay, almost at the end here. So when it comes to kinesthesia versus vestibular sense, kinesthesia is your understanding of where your individual body parts are in relation to the rest of you. So, if I close my eyes, I should still be able to reach out to the side or forward, but, to what degree am I aware of where each of my individual body parts are in relation to the rest of me? Well, that kind of gets somewhat muddled when you have to do certain tasks like pin the tail on the donkey, right? Well, I know where my arm is in relation to me, but I don't know where I am in relation to space. And so that feeds into your vestibular sense, the sense of your whole body's movement and position relative to the rest of space. And your vestibular sense includes your sense of balance. So kinesthesia is just about sensing the position and movement of your individual body parts, whereas vestibular sense is your sense of movement and position for your whole body. So your whole head and body's position and movement, versus kinesthesia, individual body parts, my toes, my arm, my hand, my knee, these sorts of things. And vestibular sense is influenced by your semicircular canals and your vestibular sacs. Know that these two things, semicircular canals and vestibular sacs, have a ton to do with balance. They have a ton to do with balance. Super important. When your head rotates or tilts, the movement stimulates hair-like receptors to send nerve signals to the cerebellum, and that enables you to sense your body position and maintain balance. So that's how your semicircular canals and vestibular sacs ultimately influence balance. All right, last thing to go through. When it comes to sensory interaction, that is when one of your senses influences another of your senses. So the smell of food influences the way that the food tastes to you. So two of my senses, smell and taste, are interacting with one another in order to build my perception of what that food is like, or how I experience that food. And it doesn't just have to be, smell and taste. It can be any two senses that are interacting with one another to influence your ultimate perception of something. And then when it comes to embodied cognition, this is the last thing that I want you to write down on your worksheet. Next to embodied cognition, write down, your body is affecting your brain. So a sensation is affecting a perception. Your body is affecting your brain. So you have a sensation that is affecting your perception. So embodied cognition is when a bodily sensation influences your cognitive preference or judgment. So the experiments that we've done on this is, we give someone a warm cup to hold on to while they do an interview. And we give, another set of people a very cold drink, like an iced water, to hold on to while they're doing an interview. And the people who are holding a warm beverage actually tend to rate their interviewer as being warmer, kinder, more affectionate, more comforting than people who are holding a cold glass of something. They actually say, my interviewer was somewhat standoffish. They gave me the cold shoulder, that sort of thing. So even though there's really no reason for it to change your perception, your bodily experiences do affect how your brain perceives the world. Your sensations do affect your perceptions. Another funny thing that we've learned in relationship psychology is that if two people are out on a date and they're sitting at a table that's wobbly or unstable, they're actually more likely to perceive their interaction as unstable or their connection as insecure, something like that. Unsteady, unstable. So again, this is your body affecting your brain. You have some kind of sensation that actually influences your perception. Okay. And then we have this idea of extrasensory perception, ESP. This is a controversial claim that you can have perception without any sensation, without any sensory input. So this includes three components: telepathy, clairvoyance, and precognition. Telepathy is mind-to-mind communication. So if I am thinking of something in my head, telepathy is the idea that I can take that thought from my head and put it in your head, and then you can tell me what I'm thinking. So mind-to-mind communication. Clairvoyance is being able to perceive something happening away from you, but still within the same point of time. So like if I perceived that right now there was a house fire in Nevada, and I was correct that there was a house fire in Nevada, then I had clairvoyance. I was able to perceive an event that was happening outside of my immediate awareness, right? Outside of something that I could see, hear, taste, touch, feel, right? Something that I'm just saying, I have this sense that this is happening, and I'm correct about it. Well, then I was clairvoyant. Precognition is being able to perceive a future event. like an unexpected death in the next month, right? I think my sister is going to die in a car crash or something, and then I turn out to be correct. I had precognition. I was able to perceive a future event. And so parapsychology is the branch of psychology that tries to study extrasensory perception and other things. But really kind of the point to make on these is that anytime we've ever brought someone into a laboratory who says that they can do telepathy or they have clairvoyance or they have precognition, they have never been able to reliably show us in a laboratory setting. They've never been able to consistently get things correct in a laboratory setting. And so ultimately, even though ESP is kind of a cool idea, we just have absolutely no scientific backing for any of these things. So it's a cool idea. Maybe we study it a little more, but ultimately, if a question asks you, which of these three things is, most helpful for law enforcement, or like which of these three things is most reliable or something along those lines, the answer is always going to be none of them. None of them are reliable. None of them are proven. Okay. So that is the end of this one. Sorry, it was a little bit longer than usual. But do make sure to submit that pre-session worksheet. There are less things to write down on this worksheet than others, so do make sure that you have the 3 things written down that I did say in this video. All right, and I will see you at the Zoom session.